All right, hello everybody. So I think I made a mistake in explaining Mendel. So I'm going to create a video for you here on Mendelian genetics. I think the mistake came with the explanation of the two-factor cross. So in this case, we talked about one-factor crosses right here. Talked about a single allele that is either homozygous dominant, heterozygous dominant, big A, little a, or homozygous recessive. So in this case, again, we talked about this idea that we are only crossing two parents, parent one and parent two, in a one generational cross to seeing what they got. Both parents in this case were heterozygous, meaning large A, small a, and they were crossed. And what we found out is that 25%, which is the two capital A's, AA dominant, homozygous, is 25% frequency. Heterozygous children was 50% frequency, and 25% in the lower right-hand corner was homozygous recessive. All right, so we also talked about this idea that not all things pass genetically, pass with just one single allele, that some things in the DNA are passed on multiple alleles, or at least you have alleles that are sometimes paired together. It is far more complicated than that, as we know that things like disease and other things that affect us biologically have three factors: ecological or environmental, biochemistry, what's going on inside your biology, what is stimulating that or affecting it outside by the environment, and then last but not least, obviously your genetics. But your genetics only can contribute up to a certain percentage, somewhere in the range of maybe 50 to upwards of 75% of the factor to create a certain type of disease or disorder. You have to have certain biochemistry issues inside of your body, maybe due to diet, um, consumption of certain types of products, and then you have environmental issues as well. All right, so where I think I screwed up is I took a couple of new parents here, P1, P2, and I didn't factor them properly. So I'm going to show you how to actually factor out these heterozygous parents. So I created a new page under the evolution unit in the content library. So let's talk about genetics of two-factor alleles. Okay. So in this case, we have a parent. So we'll say P1, parent 1, is completely heterozygous. All right, so we'll say that A or A equals eye color. B or B equals hair color. Again, large B is dominant, small b is recessive. So in this case, hair color will be brown is dominant, and small b is blonde. And brown is dominant. And we know that this is going to be blonde. I'll write that in small letters. Okay, up here, eye color, dominant is going to be, again, brown eyes of certain shade. And then, obviously, this is going to be blue eyes, because they are recessive. Alrighty, so let's do the cross. Let's say we have two parents that are heterozygous. So P1 is going to be AA, B, B. Heterozygous, that means that this parent has brown eyes and brown hair, because the recessive is suppressed by the dominant always in genetics. In the case of our lab, we had paired recessives where that part of the population died off. Hence, AA, if you had two small a's, or homozygous recessive, that part of the population was dying off in simulation 2 of our Hardy-Weinberg lab. Okay, so parent 2 is also heterozygous. And what we're going to do is we're going to factor this out. So in this type of cross, to make a larger Punnett square, try to make this as even as possible. All right, so now we have to do something which almost resembles foiling. So P1 will get foiled up here, P2 will get foiled here. So what you have to do is you actually have to take this letter, the first allele, and pair it with the B's, okay? The B allele. So we're going to go A, pair it with the first letter over in this combination will be B. This A will get paired with the second allele, so A, B. Now we go to the second pairing. So now we have B is the first allele. It's going to pair over here, okay? So what we'll do this time is it'll be B, A, and B, A, like this. Obviously, the second parent down here is going to be exactly the same because they are the exact same allele, heterozygous. A, B, A, recessive B, dominant B, dominant A, and dominant B, recessive A. All right. Now what we do is we recombine them all together. And so the first child in this case is going to be dominant, 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 dominant. So obviously they're going to have brown hair, blue eyes. So in this case, a homozygous A B factor is brown. All right, next person, next child, be A and A. Okay, in this case, we're going to combine the B's. Remember, the dominant always gets written first. So capital B and small b, this child is still going to be brown and brown. Second pairing, again, we're going to combine the two letters together again, A, B, B, homozygous, dominant. And then the next factor, we're going to have A, okay, hetero, and then B, two capital B's, this is homo, this is a hetero-homo combined. All right, next factor, again, we go A, and we continue on. So what I would like you to do is try to think about combining the rest of these together, keeping in mind that the recessive gets written second and the dominant is always written first. Like up here in the upper right-hand corner, this fourth child here, even though we don't look at it that way, it is that the dominant allele gets written first, the recessive second, but the same letters always get paired together, whether it's uppercase or lowercase or recessive or dominant. They have to go together because those are going to express whatever the phenotype is going to be. Remember, the phenotype is the expressed trait. Hopefully this helps, and hopefully my video works. This is the first one I am making.